Thank you, Wu-Jing, for inviting me here. And it's very nice to be on the same program as Mariano and Josh. We all taught in this array tomography course last November in Munich, and we'll be teaching again in Buenos Aires, like Mariano just said, in sometime in November. So I was going to talk about array tomography of the kidney glomerulus, but something unexpected came up, so I'm going to talk about that. So I'm in the Department of Cell Biology at the University of Connecticut Health Center, but in the summers, I'm lucky to have a lab at the Marine Biological Lab in Woods Hole. So last summer, a new Thermo Fisher Hydra SEM was installed there at the MBL, and I was told that this was a FibSem scope, which used oxygen plasma instead of gallium and did something new called spin mill. So Louis Kerr is the director of the EM facility at MBL. And during the summer, we started trying to learn how to use this new microscope. It was a challenge because both of us had never done FibSem and didn't know too much about it. So, well, one of the great things in science is to work with someone else on a difficult problem. You bring different backgrounds and approaches, or sometimes one person is rather impatient, even reckless, and the other is thorough and pays attention to detail, but somehow their combination gets it done. And I hope Louis thinks that's funny. So in September so we struggled with that for a while, but in September, Vivek Subramanian from Thermo Fisher came and showed us how to do it. But it's still a little complicated, and we have to learn other things, such as how to troubleshoot problems. There are several Hydra in the US, but it seems that they are all being used for cryo-EM. So we haven't been able to get advice from other users. Since November, I've been coming back about once a month for a week to work with Louis, and now we are fairly confident about being able to do it. I'm going to show you the workflow and then a couple examples, and then Louis will give his perspective in the last few minutes. Okay. So you start with okay, let's see, hopefully this. Okay, so you start with a regular EM block. So it might be necessary to have it heavily stained, so we usually use Roto and embed it in EPON. And this is looking at a aluminum SEM stub, and you glue the block to the stub and trim it to about 1 millimeter square, so kind of small. So this is partly for the spin mill, which needs to have something small to work, but it's also important for charging. So charging is fatal to doing this thing, and it gets worse if you have a bigger block. And we also paint the sides with a conductive paint, so to help with that charging problem. So you want to have this, in order for the spin mill thing to work, you have to have the surface of the block parallel to the surface of this stub. So what you do is you put the stub into the microtome and set everything to 0, and then you trim the face, and then you trim the face so that it's parallel to the surface. And then, of course, you have to trim into the tissue and decide exactly where you want to start imaging. This is a challenge that every electron microscopist, is familiar with. Okay, and then once you have it, then you put it in the scope. And I'm going to hopefully show you, this is an animation of how it goes. So Uh, it's on a post, which can move around and turn and tilt. And then we're showing here two guns. So one is the electron gun, which is going straight down, and the other is the oxygen plasma gun, which is at a angle of 52 degrees. And the whole process goes, oh no, I pressed the wrong button. The whole process goes like this. So it tilts and then okay, so that's too fast. But when it tilts, then that's the milling part. And then in this orientation is the imaging part. And the way the microscope is set up, the two beams are intersecting at a point which is called the eucentric point. And so in order for this to work, you have to get the surface of the block right at that position. So so you have to do that. And then another thing is that this animation doesn't show that you have to rotate the block. This is for the spin mill, so it mills for several seconds, and then you rotate it 5 times. So 360 divided by 5 is 72. So you rotate it by 72. Okay, so that's so this next animation. Oh, let's see. Yeah, this is just to show what happens if you're not in the right place. You see, it's never going to work. Okay, but see, this is so it won't work. Okay, so this is what the spin mill looks like from the point of view of the oxygen plasma gun. So it's 4 degrees. It's it's angled at 4 degrees, and so the oxygen plasma is coming straight into the screen, and then what happens is that the block is rotated 5 times, and so that's what the spin mill is. All right, so the first part of setting up the whole thing is to get everything in place to do this, and um, so to get it at the eccentric point, you get an image of the surface, and you're at about 1000x, and then you tilt the stage. Well, okay, so this is just a screenshot which doesn't show, but you tilt the stage, and typically that spot which is underneath the cross will move. And what you do is you use this thing called jog Z. So you're going to change the Z position so that it comes back into the spot. So what this means is that as you tilt it, that this spot doesn't move. So that's to set the eccentric, and it's not too hard. Okay, now this other one, it's harder for me to explain exactly how this works, but let me explain the problem. So, we're going to spin it, the spin mill part, and you can see that this block is not in the center of the stub, and so there has to be some kind of uh, some kind of fancy correction so that when you spin it, that the beam ends up in the middle of the stub. And so I'm not quite sure how that works, but this little video shows how it does. So you again, you're at 1000, and you find a point at the corner, at one of the corners, and then it rotates by 180, and then you click on that same point, which is slightly off, and then somehow it's able to correct the rotation. Okay, so it's not, I'm, I mean, this talk is not meant to show you exactly how to do it, but this is how, this is what's involved in setting the rotation. All right, then if you got it right, then you tilt the thing. So you can see on this that I don't know. Yeah. So on the left-hand side, there's an image of the surface and then on the bottom, it's you can see that that the block has been tilted. Actually, this is minus 34, and the oxygen is coming in from the right, and it's that's at 52. So it turns out that this is a 4-degree angle on this is looking at the surface from 4 degrees, and so you want to make sure that as you turn it, so you're going to let me see. Um, I'm sorry, this is a little bit. Um, no, okay, so I don't have that in there. But anyways, as you turn it 72 degrees, this the center of the screen should be on the center of the block. So you want to and in fact, if you set up the eccentric and the rotation, then it will be like that. So, but you have to check it. Okay, then uh, if you try to image this, it doesn't it doesn't look good. So what they say is you have to discharge the surface. And what you do is you go to this panel. So this is a panel which controls the plasma beam, and you're going to set it to a very high current. So this is 64 nanoamps. Usually it's 0.14 nanoamps, and you see we're using oxygen and 12 kV, and you kind of flood the surface with this oxygen, and I think. Well, oxygen plasma is positive, and somehow some of it gets embedded in the surface, and maybe this is what's going on when they say discharge the surface. Okay, so once you've done that, then you can bring the block back to 0 degrees, and you can image it using backscatter. And so at that stage, you kind of look at what you have, and we usually use 3 keV and zero point 0.4 nanoamps, and there's something called immersion mode on the FBI scopes. Well, and of course you set the STIG and things like that. So you got to you would adjust the imaging parameters, so it's going to look good. All right, so now everything is set on the microscope, and another software is launched. So this is the ASV software, and let me see if what I have here. Okay, so now we're in this other software, and so first you have to you give the project a name, tell it where to save the images, but is next thing shows, I believe. Yeah, okay. So this is looking at the surface of the block, and you set up something called the area of interest. And so this is the area that's going to get milled. At least we think it is. Okay, so you set that up first. Uh, okay, so then you start imaging using backscatter, and you're going to try to figure out. Okay, so let me go back. So that this area is approximately 1 millimeter, but when you or this circle, the diameter of the circle is approximately 1 millimeter, but when you image, let's say that you're imaging at high magnification, at high resolution, say 5 or 10 nanometers per pixel. So what we usually do is make a 50 micron area. So you have to look in this 1 millimeter circle and find a 50 micron square area that you're interested in. And actually, you can pick a couple or two or three, but it would take way too long to image this whole area. So you have to choose a small area to image. Okay, so that's shown in this one. So it starts off, kind of, and then you kind of define an area like this. Okay. Now yeah, okay, this doesn't really show very well, but what I'm trying to do is show this panel that's on the upper right, and in that panel, which um, you can set the well, it has the area, but you're gonna set what resolution. So in this one, the second box from the bottom is we set it to 10 nanometers. And so this will get to 1500 pixels. And then on the bottom line is the dwell time. So we're often doing 3 microseconds. So 1 microsecond is a little bit noisy. And then you can set it to uh, how many images you want to get. And then um, on the right, on the next bottom, that's the thickness. Now that's something I forgot to take a picture of before, and so there's a panel which tells you how thick the milling or how much surface is going to get milled, and it turns out that that's not really calibrated, so it's it you have to make a setting. You have to choose a setting for what material you're doing, and then there's some kind of curve which they think is it. So actually, this number is not exactly. I mean, it's close, but it's not exact. So, well, anyways, that's one thing we have to work on a little bit. Okay, so we've set this. And then the next thing is you set up two areas for the autofocus and the stigmation. And also you can see this square, the big square is green. And so they have something called drift correction. So you can imagine that as you're doing all these stage movements and stuff, it's not necessarily going to end up in the same place. So what the software does is it keeps track of the previous, the first image and then each successive image and compares it with what it's seeing, and then makes a correction, any correction for problems with the stage. Okay, then there's also an autofocus. So the little orange box next to the green is the area where it's going to do an autofocus. And then the other one is where it's going to do a stig. And the auto stig is every 10th image, but the autofocus is done on every image. And it's a little bit tricky to get the right numbers in there, but we can do it. Okay, and then all right. So this is something on the bottom of the of that. Let's see, let me. Yeah, it's not showing. Okay, this is on the bottom of that screen. And what you can see is that on the left, bottom left, there's something that's been cut off, but then next is the milling. So this is actually the place where you determine, where you tell it how much to mill. And then you can see that the default is to have two regions of interest. And then once you get it started, well, let's see, okay. Well, off to the right, there's yeah, actually, but anyways, there's a button that says start job or something like that, and so that uh, uh, once you press that, then it's going to start milling and imaging. So there's also a thing called pause, I think, and then abort. So, well, okay. So first, if you start it, then it's going to, the software is very nice on this. So as it's collecting images, you can look at it in this little panel called results, which is off to the left. This little button that's next to ROI, and then you click on that, and you can see what it has done. And you can decide, for instance, that you picked the wrong place. So you pause it, and then you can change some of the things, not all of the things, but you can change some of the settings, in the middle of a run, and then you resume it, and it'll keep going. I think you can even add a new, take out say, no, I don't want this region of interest, and then add another one. So this part of the software is pretty good, I think. So it does it. Um, so uh, so I wanted to point out that the I remember we started with the stub, and we put it in a microscope, and then we do all these alignments and choices and stuff, and then the whole process starts when we hit this run button. And if everything goes smoothly, that takes about 30 minutes. So it's really not too bad. I mean, there's a lot of you know moving around and stuff like that, but it's not that bad because once you start that thing, it just goes automatically. And if everything is set up right, then you can get your data. Okay, so usually we do overnight runs, but we have done a few that take 2 days. Uh, the whole process is rather slow, so when we've been doing it, it's about 10 sections per hour. So let's say that you have 20 nanometer sections, so that means 200 nanometers per hour, or overnight of 16 hours. That's three microns deep, so not very far. If you do a day, that's 240 sections, 2.5 microns, or if you do 2 days, then that's um, uh, 10 microns. Yeah, I'm not sure if I'm doing, yeah, yeah, okay. If you do 2 days, that's about 500 sections, and that's, you're going 20 nanometers, that's around 10 microns. So that's definitely, one of the things that you have to work with this method. All right, now I'm going to try to show some examples. So let's see, I want to come out of this share. Where's my mouse? Okay. Um, all right. So I guess that's showing up. Is that the, yeah, okay. So this is a salivary gland, mouse salivary gland, and hopefully this, okay. So so, um, maybe 20 nanometer section. And uh, I think this is a 50 microns, and then we can, go to higher mag. Okay, so because I'm an array tomography person, what's so striking about this kind of data is that there are no wrinkles, and there's no garbage on the sections, and then also there's it's much thinner than what we can cut. Um, of course, if you're going to compare with array tomography, you only get one shot at getting an image. So with array tomography, you can re-image things. So that's array tomography is not going to go away, but for sure, there's something very nice about just spending 30 minutes and then starting it, and then next morning there's your data. and that it's very nice data. Um, I'm not sure if I can, but I mean, in some ways, this is not, this kind of data. Anyone who's done FibSem has seen something like this. So I don't think there's anything um, different or better about um, this. It's just that compared to FibSem, there's a shorter prep time, and so. It's all okay. So we've done many runs, like a dozen or 2 dozen runs, and many of those have gone without problem. And things can go wrong. So we've had, sometimes we have charging problems, but you can work on your block and get rid of it. One thing that we don't understand is that sometimes the surface has ripples in it. And so that's something that we're working on. We would like to mill. We want to find out how thin we can go. So we've done 20 or 10. What's involved in going thinner. We'd like to go faster. So I think if we change the AOI, that area of interest, we can go faster. We'd like to mill thicker as well because there's some projects that don't require something that's so thin. And of course, we'd like to discover something. So we have some nice things, but we want to show that we can use this microscope to discover something. All right, so that's all I had to say. Maybe Louis can take over. How does that work?
You have a question? Someone asked you if you have the whole stack to show us? Amalia just posted in the chat.
The whole stack?
Yeah.
Well, okay, so this is only 50. So the original was 200. So we've done 500. Let's see. I didn't want to put something that would, potentially. Okay, so this is something like 50 microns square, and then this is going through the 50 sections.
So Mark, are you converted now? The not
Oh,
array
no.
topography anymore.
Well,
I
I
mean,
would. It would be nice if you could do everything with FibSim, but there's plenty of. Well, I mean, for the
yeah,
immunolabeling, you can't do with the.
exactly. Yeah, yeah,
And you have to know exactly whatever you're interested in. You have to know exactly where it is, and then it have to be in the block. So you're looking at
yeah,
the surface, and you want to. So there's a.
yeah. But the image analysis is going to be a lot easier than array tomography because of the alignment is much better than
Yes.
doing the sections, right?
Yes, and it's a big pain to image in array tomography, so
Yeah, yeah.
to map the images. So there's a lot of nice things.
Okay. So does Louis want to have share screen? Does he have a PowerPoint to show or he just want to jump in?
May I have a question?
Oh yeah, we all have some chats. There's Han, you want to go ahead, then Nick can follow. Yeah.
Uh, you mean I can ask a question
Yeah,
right
yeah, yeah.
now?
Go ahead. Yeah.
Okay, yeah, Mark, thank you for our presentation. Actually, we just bought the Hydra installed like 2 weeks ago.
Oh,
I do have a lot of questions. I think most probably will contact you offline. I have two questions really quick. One is you mentioned you glue the samples to the pin and you will put it on the ultramicrotomy to cut them to parallelize to the pin. Is that correct?
I'm sorry, I didn't understand your question. So we glue it to the pin, and what's the question?
The question is like after you glue the samples, the tissue samples, the blocks to the pin, and then you will using the ultramicrotomy to cut to make sure the sample surface parallels to your
Yes,
pin surface. Is that correct?
yes.
Okay, okay.
So otherwise, the spin mill won't work, because it'll be milling something that's not parallel. Yeah.
Okay, so you using the tissue, right? You do the tissue block.
Yes.
Okay. The second question is, you mentioned like you get 10 sections per hour when you do the
Yeah.
scanning. So may I ask like the size of the section, which like how big the area you're scanning?
Yeah. Okay. So that would be a 50 micron square. So, say, well, it depends on what pixel resolution and stuff like that. But say 5,000 pixels,
Uh-huh,
5,000 pixel square, and then two of those. Okay.
uh-huh, uh-huh.
And, it also depends on your dwell time and stuff. But very roughly, we're doing, 10 sections per hour. But you could,
Okay,
if you do a small area, you can do, yeah. So we're right now that's something. It's an important thing to be able to go faster sometimes. So we're that's something that we're trying to figure out how to uh, you know speed up in some
okay,
cases.
okay, great. So you mentioned like your section was like 20 nanometers.
Yes.
Okay, I just want to make sure that's number correct. So yeah, okay, so that's usually 20 nanometers is what you do for, or like this is the extreme you go.
Usually we're doing 20 nanometers. We've actually we didn't understand how I mean, that's another thing that we still have to explore. So we haven't, but yeah, the guy who came and showed us how to do it started with 20 nanometers, and so we've been just kind of doing 20 nanometers.
Okay, for sure I have lots of questions. I keep it for ask you off the line. Thank you,
Good.
Mark. Thank
Yeah,
you.
yeah.
Thank you.
Okay, Mark, there's another question. I don't know, you want to answer or Louis can answer? It's the
Yeah, Louis
size.
should. We should make sure Louis has some time. He has some other things to say, so we should make sure
Okay,
that. Yeah.
Louis, you want to pick up the answer the question? The typical size of the volume that's coming from Nick from Yale.
Yeah.
Uh,
Well, I think that Mark really just answered that pretty much. And pixel size, it depends on the resolution that you're trying to achieve and the time to do the experiment.
Yeah, okay. So can I ask question directly? So we don't have a plasma, but we do have a gallium-based FIB, right? So the question, one challenge is the size of the. I think Mark answer is about 50 micron X and Y to start with, right? Mark,
Yes.
am I right? So that's you're comfortably. So how long can you run? And usually that's Z. How long you gonna be that stable? You can run multiple days up to a week, or that's how large the data set you can get, right?
We've gone a little bit more than 2 days so far, and then we
Okay.
stopped the
Okay.
process that
So
potentially could have gone longer.
so the best pixel size you're talking about 20 nanometer, right? Isotropic pixel size. You match it, right? X and Y and Z.
Well,
XY is usually 10, and then the Z has generally been 20.
Got it. Okay. So that's my question. And so its system is very stable, and I understand you're using oxygen. Have you tried other gas, or this is the best for biological samples?
We're sort of following Thermo Fisher's suggestion that oxygen is the best for this particular technique. But we're very interested in trying to explore some of these other gases. So with the PFIB, it comes with 4 different gases.
Yep.
Uh,
Okay. So last question is, have you compared the result with the PFIB and the traditional gallium-based FIB in terms of quality of data set?
I looked at one data set, so it's an N of 1, and we could do a larger area with the PFIB, I think with equal or better sort of resolution and quality of the image.
Yeah. So actually, your quality of image looks very good. I'm just wondering if you reduce the pixel size how enough, right? So usually, typically, people do around 8 to 10 nanometer. We do isotropic the pixel size S, Y, and Z, right? So yeah, I think it's still going that way, but it's a huge potential. Yeah, that's all from me. Thank you very much.
All right. So I'm going to share my screen here.
Okay.
So I'm going to, all right. So I'll get out of the the I thought this was all set up.
You can just run from these.
Okay.
We have 3 minutes, a little bit less.
Okay. All right. Sorry. All right. I'll just go from here. Thank you. I'm Louis Kerr from the Marine Biological Laboratory, and I direct our imaging services. For those of you who have been to MBL in Woods Hole, hopefully this image brings back some memories. MBL has been in existence since 1888, and we try to do fundamental research and hands-on education, which we've done from the very beginning. And we've sort of coined the term that's our convening power to combine those two things. We currently tackle several different issues. We have a breadth of research that goes on, including environmental science, ecology and climate effects, microbes from the ocean, the planet, and human health. We also do neurobiology, cell biology, developmental biology. And then we've got two principal sort of initiatives going on. One is developing new model organisms, including this squid. And then the other is our imaging initiative, which has essentially been going on for a long time of developing new technologies there. The MBL has a research component. We have about 27 research faculty, which includes about a total of 180 scientists and support staff. And then we have a visiting science program called the Whitman Center, and that brings in about 300 people a year between the scientists, fellows, and their staff. We also have a very strong educational program. So this was from 2023. We had over 20 courses, close to 500 students. Ironically, we actually have more faculty than students. So my guess would be we probably had about 800 faculty that come in to teach the courses. And that is somewhat makes it unique. It's also very hands-on wet bench based. And this is, again, from 2023, I think. And it just shows the breadth of our courses. I want you to key that we have several microscopy-related courses.
Oh.
That is.
You want to quickly finish?
Oh, sorry.
Yeah.
And then we have our central facility. We were lucky last year to get this Leica Stellaris 8 STED and the Thermo Fisher Helios 5 Hydra PFIB, which is what Mark was just talking about. It was procured with funding from the Massachusetts Life Sciences Center. If you're in Massachusetts, I highly recommend that you become familiar with that program. If you're not in Massachusetts, I think it would be worth looking at and seeing if your own state or if you're out of the country, your area has something similar. So our Helios, this is a sort of long-term view of it. We can do standard SEM, EDS, EBSD, PFIB. We still can do array tomography, and it has a cryo stage. We have yet incorporated exactly all of those, and it somewhat might be unrealistic to think we really can. But we've been keen on the spin mill technique, and especially with Mark's contributions and interface. I appreciated his comments at the beginning of his talk. I'll let you decide who was the calm one and who wanted to just push ahead. I'll end, this is my last slide. And this is a nice image of one section, so to speak, from Jen Morgan's lab. This is a axon in lamprey. But these are some of the challenges. Mark covered a bit of those as well. We're finding that partly because it is such a new thing, the spin mill, that there's not a lot of people to go out and ask for advice. We have been talking to a couple people about trying to put together a users group and maybe doing a workshop at some point soon to try and get us all going further than we are. We're still working on cost and scheduling structures because it can do very long runs. But it's exciting times. We're also trying to figure out how exactly user-friendly it is, so to speak. Mark did a great job of running through the process, but we don't know whether we're going to have to really have staff manning it or letting people free to use it. And I'll end there. Thank you.